I'm talking into the empty stand. I'd like to get started to just let all the families in the audience today know that please reach out to us at the UMDF. We have some resources that your families may really benefit from. If anyone wants to connect to another family, we can make that happen. I also encourage you to join the Leigh Syndrome Family Network Facebook group. That's another place where Leigh syndrome families can gather. We hold probably biannual Leigh syndrome virtual support meetings, and I'd love to hear any other ideas on ways that the UMDF can support our Leigh syndrome families. So without further ado, I think it's time to get started with some Q&A. I will kind of just address the audience directly. Is there anything specific that anyone kind of thought of?
In addition to Mito, I also study urea cycle disorders, which is another rare disease that has unmet needs. And so we've had a natural history study for the last 18 years. And you're right, it's really important. And, you know, we were able to leverage that natural history study to get at least 2 new drugs approved for the community. So, and again, you know, it's kind of a pain, and our natural history study in the beginning had so much, and then we scaled it back to what we thought was necessary, and you have all this data, but, you know, the FDA really wants that. And drug companies really want it. I mean, they're like knocking on our doors because they know that we have, you know, several thousand patients, that we've collected longitudinal information, you know, for some of them who started at the beginning in 2004 with our study all the way through. So I think that, you know, the promise is the development of new drugs, and also understanding the evolution of the disease too, right? So it's really something that you do for your own individual family, but also for the Mito community at large, because the benefits that come from that will be enormous. So.
Yeah, and again, when you're on in that worst day in the ICU, we want the information about the prognosis to be accurate that the teams are accessing, not scary and historic and, you know, inappropriate. So this is another way to do it. And the other thing I would say is like what Andrea said, is having the natural history study actually attracts companies to develop drugs. So I have a lot of drugs that I think could be in Leigh syndrome, but they see it as hard. Right? They see it as, you know, there's a lot of barriers for the company to come into and do. So if we can lower those barriers and make it really clear where the need is, that attracts companies, which we need. We need those partners.
Thank you. I actually have a question that many families ask me. What do you think is the next big thing for patients with Leigh syndrome? What will it be? And what can they be looking forward to? And how can they support you all in your efforts to get there?
So right now, the PTC study isn't really for Leigh syndrome, right? It's for epilepsy. And so that's coming. There's another study coming also for epilepsy, kind of a nucleic acid that should be opening this in 2022, I think. I don't think there's any sites open yet, but I think that's coming. There's a drug from Europe that's coming. That's more in the antioxidant class. We were close to having rapamycin, which is a drug that has equipoise. It might help, it might hurt. And so the company actually, between the pandemic and funding crises, right, that wasn't able to move forward. So that's another example where, you know, we have a great therapy and we don't have a commercial source of the compound. I think there should be, continual trials, you know, that the family should be on a therapy. They should be on it for a time, maybe on an open access for a time, and then there should be on the next drug. So I think this is a disease where we continually need to put people on therapies until we treat all of their problems, right? And make things better. So some of the therapies that are coming in the adult world are for things like energy, fatigue, exercise, all of that matters in Leigh syndrome, you know, all of it. There's a couple of drugs coming into they announced here, the Cyclarian trial, which is coming for MELAS. Why MELAS? MELAS is easier regulatory wise, right? It's still brain involvement, and they have a brain penetrant drug, which is super cool, right? But it's all one gene, and they're adults, right? So we would love to see that in kids with Leigh syndrome. So I don't think there's just one therapy, and I don't think we should necessarily put all of our eggs in one basket. I think we should be testing them all, right? And that's the other thing that I think we really think a lot about, is thinking about outcomes that will really tell us if the drug is working, but we don't want to miss it. Like if we're going to look for seizures, but it actually makes kidney function better, or it actually, there are some kids with, renal tubular problems, or if it makes energy better, or if it makes vision better, we want to help all of these problems. Most mito patients have 16 major medical problems. So gene therapy, I think, is possible. I still don't know that it'll treat all 16 of the problems. Protein therapies are possible, cell therapies are possible. And I think what's really neat is that I don't think in rare disease, we have to be thinking in the hundreds of patients in each trial, and everybody can talk more about that. I think 20, 30, 40 patients is enough. Depending on if we get our acts together, have the natural history study, design things right, think about crossover studies, think about the right period of time. But if we have, as a community, come together, we work together, the families are of the mindset, yeah, we're going to keep going until we find cures, and we're not looking for incremental, 5% better. We want 50% better. We want 80% better. We want this symptom gone. I think we need to raise our sort of standards, and we need to get ourselves ready. And it's already happening, right? At this meeting in 2015, there were like no companies. Right? There were no trials. And the first trial was horrible that we participated in. It had 400 exclusion criteria. You couldn't get in, right? Like you were at the door and you couldn't get in. So I think we've all taken a much more active role. And I think the community's taken a more active role. So I don't know if that answers your question, but I think there's a lot coming.
Do you want him to? Yeah.
I'm actually good for time, but so I'm going to take a little bit of a different approach. Earlier this week, when I was flying out to Phoenix, there was an article in the New York Times about a T-cell treatment for cancer. They looked at patients with stage 4. I mean, the cancer was in every organ of the body, and 100% of the patients responded. I think there were 17 patients in the study. So the answer to that question is we don't know what's going to work, and we don't know when it's going to happen. So we have to be participating. And, Phil Yeski and I were talking about this morning about the COVID fatigue, that this pandemic has exhausted all of us. So we have trial fatigue, we've got doctor visit fatigue, we've got doctors are fatigued. We got to put all that aside, because we're not going to get to this solution until we basically all agree that we're going to solve it together. So I'm going to say I don't know. But that's not.
So on a more optimistic note, so I think consideration of trial design, right? So we've been talking to the FDA a lot for our other disorders, and innovative trial design, because as Marni said, the numbers, the N, the statistics are difficult to have an effect size, with a rare disease. So, one type of trial that was previously only exclusively done in cancer are those basket clinical trials. So what that means is you can take patients with different conditions. So you could look at MELAS and ELHAN and, Leigh syndrome, and use the same drug and have some common endpoints and some divergent endpoints based on the features of the disease. And that will speed things through clinical trials. So you can test different subgroups of populations. You don't need as many patients because you have a lot more measures. And so the FDA has sort of been, tinkering with the idea of alternative trial design. So that may help in rare disease as well.
So I think it's interesting. You ask one question, you're going to get 4 very different answers. So, I think a lot about this question. What is the next big thing and how is it going to happen? And like Dr. Gropman, I also have another life where I work with a different rare disease. And I think with Leigh syndrome, I think a lot about the blood-brain barrier. And we've got all these great therapies that are being tested that don't cross the blood-brain barrier. And Leigh syndrome, great, your kid's hypotonic, there's a problem with their muscles, okay, they have some kidney issue, okay. You can't live if your brain is damaged. The brain is the most important part of your body, hands down. It has to work. You're a neurologist, you agree, has to work. So we need to find a way to get things into the brain. I believe it's an inflammatory process that's happening in the brain. We need to get that inflammation down. I don't know that, but that's what I believe. And so I think we need to find something that can cross the blood-brain barrier and affect that inflammatory process. And I'm being intentionally vague. In my experience in medicine, it's kind of like what Dr. Cohen said. We don't really know what the next big thing is. It always just appears. And when it appears, it often appears very quickly and moves very rapidly. And we just need to be ready to grab it and move with it. I think a lot of people have focused a lot on gene therapy. And I'm really excited about gene therapy. I've been excited about gene therapy since I was in medical school. And they have been really close to getting it right since I was in medical school. And I believe that as soon as someone figures out how to do it, it's going to happen, and it's going to happen fast. And it's going to be translatable to all of the different single gene disorders. So I don't necessarily feel like every single gene disorder needs to be focused on it. Because I think that once it happens, it's going to happen, and it's going to be very rapidly translatable to everybody. But I also look at a lot of you in this room who have children with Leigh syndrome, and I'm assuming that they've already developed symptoms in their brain, and reversing their single gene disorder is not going to take those symptoms away. And so we need to also find ways to treat the brain and what's already happened in the brain and what's happening to the brain. And that's my opinion.
Thank you, everyone. Any other questions in the audience? Steve?
Yes, it has brought down a lot of barriers. So the FDA has a lot of barriers, and these orphan drug classifications, they are way bringing them down. So I know it seems like it's not, and it still seems like a very cumbersome, time-consuming, and difficult process. But if there was not the orphan drug status, it would be even worse.
Sean, I saw you had your hand up.
Yeah, so it's been 25 years, I think, or so since PGD existed for nuclear gene disorders. And I can't remember the name of the little girl where it was first done, but yeah. So, and some labs got expertise. That nuclear gene change, right, is consistent from cell to cell to cell. So it's not there or not there because of this issue of heteroplasmy and the liability that goes with being accurate, and how certain are you? A lot of the methodologies that people were using just weren't sensitive enough to on such a teeny tiny amount of DNA to be sure. And so people were trying to do it, and it just wasn't accurate. And there also wasn't knowledge about how low do you go where you can counsel and be sure. If there's any present, it could become enriched, right? And so what's the likelihood of being enriched? At the other side, how high can you go? And so Bobby McFarland, they're going up to like 30%. In some cases, it depends on the mutation. Some mutations at 20% are horrible. Some are tolerable. So some of it is the knowledge. Some of it is the technology. There haven't been, so in America, it's a very litigious society, and there are no labs, no good labs that would take on sort of the risk of saying they won't do amniocentesis or chorionic villus sampling either for mtDNA to be able to predict, because they don't want to be, it's a very tough issue here, and they don't want to be responsible, for getting it wrong. And what's remarkable also is that a lot of the work out of England has shown that the day 3 embryo mtDNA level in several of the cells, one, it's representative in all the cells, number one, and two, it's much more representative of the levels we believe in the baby when they're actually born, much more so than an amnio or CVS, later in pregnancy. We're still trying to get them to do day 5 embryos. Right now, all the data is available for day 3 embryos. So we're going to approach that next. But part of it is the technology. And believe it or not, that took a lot of effort to figure out the right, Shrikant Metalapoff in America figured out a lot of it, but he's only a research lab. So the only other lab that did it because of MRT was the Newcastle lab. And so that's who we're using. And then they weren't accepting samples from around the world. So that's something that we've just been able to work on with them and successfully figure out. I don't know that anybody's invested in doing it right. I still think it takes a lot of expertise. I think it can be done. I think somebody has to really commit to it.
Thank you. It's time for us to wrap up, and I want to invite a special guest up here. Lori, will you join me for a second? I hope you all know this person, but it is because of her that we're here together getting this great information. She has been a great partner with the UMDF in many, many ways. And I just want to thank you. And I just want to hear how this idea came to be and let everyone know how we made this happen today.
Thank you. You're just embarrassing me. But thank you, seriously. So I have a 13-year-old son named Will who does have Leigh syndrome. And in full disclosure, our doctor is Dr. Koenig. And so a couple of years ago, after he was diagnosed, our family and friends were like, this is not acceptable. We are not going to just take this diagnosis of death and not do something. And so over the last couple of years, we've formed a nonprofit called People Against Leigh Syndrome or PALS, and we have worked closely with the UMDF. We're part of the International Leigh Syndrome Roadmap Project, which we feel like is really important. We've supported grants with the UMDF. We've done some grants of our own. We support the trials registry that Dr. Koenig runs. And I have come to the conferences now for, I don't know, I'm kind of a groupie, I guess. I've missed one because I had a daughter. And I just always felt that we needed this. And it had evolved over the years. And I know Heather Thornberry was instrumental a couple of years ago in doing some patient meetups. And I just felt like we as families needed to hear from these guys, the real education piece of what's happening in the brain and how does it really work. And so I just bugged the UMDF every day, every year. And I was like, we need this. And so I'm just really happy to see it come together. So thank you guys for everything that you do for our families, and our kids would not be where they are without people like you guys. So thank you.
Thank you for closing that up for us, Lori, and thank you to our amazing panelists today. I learned a lot. I do want to let everyone know that the slides will be made available to you after the event. We do believe there's a way that you can download them from the app if you go on your computer, but we have to test that first. But if that is not possible, please just reach out to us, umdf.org. You can find the Get Support form and fill that out, and I will make sure that you get the slides. I understand why you'd want them too. They were amazing. I want them. So thank you for joining us. And hopefully we will see you in Charlotte, North Carolina for Mitochondrial Medicine 2023.